Dear students, welcome to this lecture where we are going to continue our conversation on functional diversity and focus more on microbes that interact with elements that influence our nutrient cycles of earth. Of course, when it comes to carbon cycle and water cycle, almost every microorganism we know has contribution to it. But today we are going to focus on sulfur and nitrogen. So first we will start with the sulfur cycle. Now instead of focusing on sulfur cycle, let us look at what microbes do with sulfur and how they can use it. So I have written here different forms of sulfur that are relevant to most microbes that involve with either sulfur oxidation, sulfur reduction. So first we have sulfate here. It is the most oxidized form of sulfur that is very relevant to most microbial communities. There is another higher form of oxidation for sulfur, but this is pretty good enough for now. So this sulfate, it will be, when it is reduced by microbes, they get energy and they reduce it in a series of different enzymes, and we will see about them very soon. So microbes can reduce sulfate to sulfite and further to elemental sulfur, and then to hydrogen sulfide, and then hydrogen sulfide, which is the most reduced form of sulfur. So at each step, as the microbes reduce this sulfur, they generate energy which they can use as source of energy. There are few microbes that can actually go all the way from sulfate to H2S, but usually it is a consortium of microbes. It is a group of microbes that get together and participate in this reduction of sulfate. So the arrows show the reduction pathway for sulfur. There is another parallel pathway which is the oxidation pathway. So there are microbes that we have which oxidize elemental sulfur. So this is the oxidation pathway, and as they oxidize it, they generate energy. Then we have microbes that oxidize HS minus and they generate energy, and we have microbes that oxidize hydrogen sulfide to generate energy. Now earlier I mentioned that because of the principle of thermodynamics, chemical reaction will be energetically favorable only in one direction. So an obvious question that must arise now is, why does sulfur particularly lend itself to bidirectional movement of electrons when energetically only one direction should be favorable? Now this is a very good question, and I want to mention why. Under normal circumstances, this direction where reduced form of sulfur are getting oxidized is more energetically favorable. However, under extreme anaerobic conditions, when no other electron acceptor is present, then microbes invest in energy to reduce sulfate so that they can oxidize their electron donor and make up for the energy. So the energy that they get from the electron donor, so part of this energy is used in reducing sulfate, and still there is a pretty good chunk of energy left that microbes can use for their day-to-day sustenance and for their growth. And this is how microbes make use of, under extreme distress, they can reduce sulfate and sulfide. So now let us take a look at the different kinds of microbes that have to do with sulfate reduction or sulfur oxidation. So last class we talked about green sulfur bacteria. This is where I ended the lecture. Now it is very important to note that green sulfur bacteria and other bacteria that we have talked about before, like purple sulfur bacteria, they store sulfur as an energy reserve, and then they cannot no longer carry on phototrophy, but their primary source of metabolism, primary source of energy is not sulfur oxidation or sulfur reduction. When other bacteria such as green sulfur bacteria and green non-sulfur bacteria, these are similar examples of phototrophs that we covered in the previous lecture, and they do not metabolize sulfur as their primary source of energy. So green sulfur bacteria, let's talk about them a little bit in detail. This is a picture of green sulfur bacteria, by the way. They are thermophilic, they love heat. So where will you find them? In hot water vents, in hot springs, and under sea volcanic vents. And if you see, well, in this particular slide it is not very clear, but there are the chlorosomes which contain their pigment, photosynthetic pigment. They are usually at the cell periphery, and they can be blue and green carotenoids, blue and brown carotenoids. Now green non-sulfur bacteria, they underlie in these particular three genre. They are anoxygenic phototrophs of chloroflexi, and they are widespread in most environments, and I have sequenced many environments, and I'm more often than not always find some chloroflexi, and most of them have not been cultured in isolation. That's why very little is known about them. So one of the initial ways in which we tried understanding the metabolism, the function, and the genetic fingerprint of a microbe was we would grow them in lab. That's called growing in culture or culturing them, and once we have grown them in lab, we would study them, how they look like under microscope, what kind of food they eat, how they eat, what proteins they translate, and how they utilize them, and what their genetic signatures are. But many microbes we have not succeeded in growing them in culture, and chloroflexi are them. So the only way we can know a chloroflexi exists is by, without culturing them, sequencing their genes and then finding it out. There is a particular chloroflexi, thermomicrobium. It has very unusual lipids. It has neither ester nor ether linked chains like bacteria or archaea, and when it has to make bilayer, it has two di-alcohol molecules that oppose each other. So there is an alcohol molecule here, there is an alcohol molecule here, and they have hydrophobic chains and they just oppose each other. That's how it makes bilayer. Chloroflexes were first found when in alkaline hot springs, and they had made thick microbial mats. The other phototrophic bacteria that are similar in line with what we were talking about in previous lecture are Heliobacteria and Acidobacteria. There is some information here that, and let's go through it. So Heliobacter, the key genera are Heliobacterium and Chloroacidobacterium. They are phylogenetically coherent. Again, this implies that they are only found in certain phylum, and that is Firmicutes. All of them are Firmicutes, and they are gram positive. So if you are gram positive, you lack that periplasmic cell membrane, and the first interaction of the gram stain would be with your cellular membrane. They are anoxygenic phototrophs, and they have a unique pigment, bacteriochlorophyll G, and that's the reason why we wanted to mention them separately. And they are phylogenetically very restricted. They only fall under Firmicutes, and they can only utilize a narrow range of organic compounds as their carbon source, and they are morphologically also very similar. They are either rod shaped or they are filamentous, strict anaerobes, and if they do not have light, then they can shift to chemotrophy. So they are not obligate phototrophs. And then we have Acidobacteria, which are anoxygenic phototrophs that were first discovered in Yellowstone National Park. So now let's come to microbes that not only store sulfur for in case they have to shift to chemotrophy, but they actually use sulfur compounds as their primary source of energy. And that is where what I mentioned at the beginning of this class about sulfate reducing all the way to hydrogen sulfide, and also hydrogen sulfide, HS minus, and sulfur, elemental sulfur getting oxidized and serving as a source of energy for microbes comes into play. So let's look at this slide. We have sulfate here, and we have three different enzymes mentioned here. By the way, there are multiple steps here that have been all generalized to one straight arrow. They consume one ATP and release a diphosphate, so ADP, and these three enzymes, they at different steps they catalyze this reaction, and eventually sulfate it gets attached with adenosine 5 of the ATP, and that's why diphosphate has been removed, and then with other series of enzymes it gets reduced to sulfite, and then in this some sulfite to trisulfide, and then hydrogen disulfide. And in this process, in sulfite reduction, there is a series of enzymes that are dissimilatory sulfite reductase. I want to talk about dissimilatory and assimilatory enzymes. So dissimilatory processes are the ones that do not assimilate the element that they are dealing with. So in this case, dissimilatory sulfite reductase will reduce the sulfite but not make sulfur in sulfite an integral part of the cell. We are only carrying out this reaction so that sulfite can reduce and we can get the energy. So the energy will become integral part of the cell but not sulfur itself. Now one other reason why I want to mention this DCRB, DCRB, DCRVB, DCVA, DCRA and DSRC, because these serve as markers for sulfite reduction. Remember, I was telling you, if I know the function, I can know what kind of environment I am dealing with. And then once sulfite has turned into trisulfide, it further can be reduced to hydrogen sulfide using another enzyme. Alrighty, so basically sulfate turns into sulfide to bisulfur, and it can also go vice versa. So sulfide is not very long lasting, because the moment it comes in contact with any electron acceptor, it's a very good electron donor. So it comes in touch with an electron acceptor, it's likely to turn into elemental sulfur. So this reaction is pretty quick. Alrighty, so we have three different kinds of microbes that use sulfur. We have dissimilatory or dissimilative sulfate reducing bacteria. We have dissimilative sulfur reducers and dissimilative sulfur oxidizers. So the ones who reduce sulfate to sulfide and maybe to sulfide, and then those who reduce sulfur to sulfide, and then those who oxidize sulfur to more oxidized forms of sulfur. So first let us look at dissimilative or dissimilatory sulfate reducing bacteria. The panel on the right is a dendrogram of certain dissimilatory sulfate reducing bacteria, D-sulfobacteriaceae, D-sulfovibrio, and some firmicutes, D-sulfotomaculum, and so on and so forth. And the way we have generated this phylogenetic tree is by looking at the enzymes that are active, DCRB, mostly DCRB here, L2, LC3, and we have an extended version of this tree also includes DCRA. Now this tree here is shown to express to you, to drive the point home, that dissimilatory sulfate reducing bacteria are immensely diverse. They are found in proteobacteria, in nitrospira, in firmicutes, thermodisulfobacterium, and eukaryota. Now let us look at sulfur oxidizers. So these will oxidize your sulfur to higher forms of more oxidized forms of sulfur, such as sulfite or sulfate. And sulfur oxidizing bacteria are gram negative rods or spirals. So you can see morphologically they are pretty limited. So and they grow in filaments, and they obtain energy through oxidation of reduced sulfur, which includes hydrogen sulfide, elemental sulfur, and thiosulfate. So they can reduce all these forms of sulfur. So these are sulfur oxidizers, and sulfur is not necessarily elemental sulfur. Molecular oxygen serves as terminal oxygen electron acceptor, and this produces sulfuric acid. So if oxygen is present and the reduced forms of sulfur are being oxidized, then we will get sulfite and sulfate. And if there is water present, definitely this is sulfuric acid. As I mentioned before, there are some compounds that are more oxidized form of sulfur, and there are some that are more reduced form of sulfur, but this is the general chain. Then we have thioploca. These are two examples, Thiothrix and Thioploca. Thioploca, the picture is here. They are large filamentous sulfur oxidizing bacteria that form cell bundles surrounded by a common sheath. So if you look very carefully in this particular picture, you notice that there is a filament, a green cylinder kind of filament that is going diagonally through this picture. Now this is not one cell. Don't think that if you see a long thread of Thioploca, it's not all one microbe, but in fact this is one common sheath that surrounds multiple cells. So the cells grow on one axis at top of each other, and there is a common sheath that runs. So morphologically we might get an impression that the cell is very long, but it's just a common sheath that covers these cells. These thick mats of this Thioploca are found on ocean floor near Chile and Peru, South America, and they often couple with anoxic oxidation of H2S with reduction of nitrate to ammonia. So their electron acceptor is often nitrate, not oxygen. Thiothrix, now this is Thiothrix, these are filamentous sulfur oxidizing bacteria, and here the filaments hold on one end. So in one end they hold on to each other and they form cellular arrangements called rosettes. So these are like a bouquet. So at one end they are attached to each other, and then from that end the filaments come out. These are obligate aerobic mixotrophs. Alrighty, now these are non-filamentous sulfur chemolithotrophs. So they are non-filamentous, they don't make filament, and they are sulfur chemolithotrophs, and you can notice here these small vacuoles where sulfur is either being stored, and these are filamentous sulfur oxidizing bacteria. By the way, if you see a name mentioned to the right side of the picture, it's the name of the scientist who took the picture. Alrighty, now this is a picture of giant sulfide oxidizing bacteria, Thiomargarita. Now these are very big. Each diameter here would be 100 micrometer. Now think about it, how can 100 micrometer serve as a good diameter for a cell to live? One of the first concepts that I covered in this class was there is a suitable size range, and the higher surface area to volume ratio a microbe has, the more evolutionary fit it would be. So why would a microbe like Thiomargarita go to such high size such as 100 micrometer? The simple reason is, the answer is given to us by these pictures. So if you look at these pictures, here we have stained the cytoplasm in the one that is black and green, and what you note here is that in the first panel, in the middle row, you note that the cytoplasm is available only at the periphery, and inside, what is going on inside? It's a big vacuole. So basically this is a cell with a big vacuole inside it, and this is a vacuole, and thus its surface area to volume ratio is still evolutionary. From evolutionary perspective, it's still fit, and this vacuole is where it stores its sulfur, because it has to oxidize sulfide, so it stores all the it stores its electron donors here. And this is one of the situations where getting a morphological insights via microscopy helped us understand the metabolism of the microbe. Now if you look here, this is the vacuole, the gray portion, the yellow portion is the cytoplasm, and then the vacuole is breaking into 4 different vacuoles, and they are showing different processes of life in Thiomargarita. Alright, now let us come to nitrogen cycle. Now in nitrogen cycle, we have talked about nitrogen cycle in detail, so you should remember some things about nitrogen cycle. And one of the first thing that we will talk about nitrogen cycle is nitrogen fixing bacteria, because as we have mentioned, nitrogen fixing is the most energy consuming part of nitrogen cycle. So let us revise nitrogen cycle first. Okay. So as we mentioned in one of the previous lectures, nitrogen cycle proceeds this way. So let us start with nitrogen fixation, the most energy consuming. So nitrogen gas that is abundant in our atmosphere is fixed into organic ammonia, and this happens via 2 routes. Either it can be through lightning, so extreme electrical charge, or it can happen through nitrogen fixing bacteria, and we are going to talk about these bacteria. So that is the microbial contribution in fixing nitrogen. And this organic nitrogen can undergo ammonification, where it degrades into ammonia, and this ammonia can be reassimilated into organic nitrogen. So this cycle goes both ways. So this is cell decay, ammonification, and then when ammonia is assimilated into organic ammonia, this is cell growth. Now this ammonia usually undergoes nitrification, aerobic nitrification to nitrate. Now the reason is why we are skipping nitrite is because this is not stable. If oxygen is present, it wants to oxidize this all the way to nitrate. Oxygen is a very good electron acceptor. Sometimes however, we will have some nitrite, and this nitrite is the reason why people get blue baby syndrome, why children get blue baby syndrome disease. And once it has made nitrate, the nitrate can undergo anaerobic denitrification. So there is no oxygen present, it is getting reduced all the way to nitrogen, which escapes us in the atmosphere, or it can continue with nitrogen fixation. Now few years ago, there was a new discovery where we found out about NMOX, which is anaerobic ammonia oxidation. Now in anaerobic ammonia oxidation, we found that ammonia can be anaerobically oxidized right away to nitrogen, and skipping the steps that are nitrate and nitrite. And nitrite can also be anaerobically reduced to nitrogen. So the way NMOX works is, ammonia interacts with nitrite and then it forms the nitrogen. Now this reaction is very simple, so let us write it down. So the way it works is, we have ammonia, ammonium, MRSA, and it interacts with nitrite. So this is not the most oxidized form of nitrogen, and this is not very stable. If oxygen is present, it will automatically go to nitrate very quickly. It is only in rare situations when we still get nitrite present in water or in our environment. Now this nitrite, under anaerobic conditions, it can combine with ammonia to form nitrogen gas and release water. Now this is not a chemical reaction, this is done by microbes, and these microbes are called NMOX, anaerobic ammonia oxidizing bacteria. Anamox, this is correct, anaerobic ammonium oxidizing bacteria, and archaea, both. So, this is a biological process and this is the reaction that happens in anamox. Now, if you look here, most of these cycles have microbial components. Even nitrogen fixing is carried out by microbes. Nitrogen assimilation into organic ammonia from ammonium is also a microbial process. Its decay is also a microbial output. Then when ammonium degrades into nitrate, this can be chemical because it is energetically very favorable. So, it does not require the enzymatic activities of microbes, but anamox, which is when nitrite and ammonia combine to form nitrogen, is microbial process. Alrighty, so let us look at nitrogen fixing bacteria. The one that we are going to talk first are diazotrophs. So, diazotrophs, as zo is an old name for nitrogenous compound, di means two. So, diazo means dinitrogen, which is nitrogen gas. So, diazotrophs fix nitrogen gas into ammonia that can be assimilated as nitrogen source. It is an assimilative process and requires ATP and nitrogenase. Now, this is energy intensive process and that is the reason why it requires such high electric charge as is available only in thunderbolts. So, it requires plenty of energy input and it requires specialized enzymes that will lower down the energy barrier, and thus we do not require 1 million ATP, we require only few ATP and we are able to and diazotrophs are able to assimilate nitrogen. This nitrogenase is a very interesting enzyme because it is very sensitive to presence of oxygen. It is irreversibly inhibited by oxygen. So, if it interacts with oxygen, if there is any oxygen present in the environment and nitrogenase is exposed to it, it will be irreversibly inhibited and we will have no more nitrogen fixing. And thus, there is immense microbial diversity that to prevent it. What it implies, what this sentence implies is that microbes have evolved in a very diverse forms to protect nitrogenase. One of the simplest way to protect nitrogenase is do not carry nitrogen fixing activity when there is oxygen present. Now, this must give you an idea what happens when we take plants, when we uproot plants that have nitrogen fixing bacteria sticking on their roots, root nodules. They get exposed to oxygen to some degree and then nitrogen fixing is hampered. So, do not do that, do not uproot nitrogen fixing legumes. This nitrogenase is encoded by NIFH gene. So, my dear friends, if you want to find out whether there is nitrogen fixing happening or not, just look for this gene. The chemical reactions that will help you amplify this gene and help you get a detection for this gene. You can even quantify it very easily. Okay, so it encodes for dinitrogenase reductase, which is a component of nitrogenase. Nitrogenase is a very complex structure. One of its domain is dinitrogenase reductase, which is specific to nitrogen fixing, and it serves as a genetic marker. More than 30,000 unique NIFH gene sequences have been found. So, just imagine, we admit every time, and we have admitted this at the beginning of this lecture, that microbiology is still in its infancy in sense that their diversity of microbes has not been tapped yet. And even then we have such a high number of different kinds of NIFH genes that we have noticed. So, note here, nitrogenase is a very, very important activity for life. Every cell requires nitrogen. In fact, each of our nucleoside is nitrogen rich. So, our genetic material requires nitrogen, our proteins require nitrogen, life requires nitrogen. And how do we get nitrogen from the abundant nitrogen in the air? By nitrogen fixing. Thus, nitrogen fixing is a very, very important part of ecosystems. And it so happens that the nitrogen fixing protein nitrogenase is sensitive to oxygen, and thus it is essential for life to continue on earth that this protein is protected from exposure to oxygen. And no wonder life has in its panic mode come up with so many diverse forms of NIFH genes and nitrogenase and ways in which it can protect nitrogenase. Horizontal gene transfer is common, and thus phylogenetic trees based on these genes are not reliable. Okay, this is one time when I need to talk about how phylogenetic trees are made. And why it is important? Because NIFH gene is commonly transferred via horizontal gene transfer. And now let us look at how phylogenetic trees are made and what is meant by this sentence here, that the phylogenetic trees based on these genes are not reliable. So dear students, phylogenetic trees are trees that we make, they are basically cluster dendrograms that we make on basis of genetic similarity. Now if you remember, the entire genome of a microbe is very long, and it does not make sense for us to classify the microbes and their similarity and their dissimilarity on basis of the entire genome, because there is so much diversity and so much different kinds of genes and genetic elements present in the genome of any microbe. So the way we go about making phylogenetic trees is we choose a gene of interest that is present in all microbes that we are interested in. Let us say I am interested in all bacteria, so I will choose a gene that is present in all bacteria. So first point is, when we make phylogenetic trees, we do not go for entire genome, instead we go for a select gene. We choose a gene and we go for it. The second point is that what should be the characteristics of this gene. So let us look at the characteristics of the selected gene. First, it should be present in all microbes that I am interested in, that is, it should be universal. Second, it should not be so different in microbes that I cannot even align them, I cannot even see I cannot even start measuring their similarity and dissimilarity. So there must be some conserved domains. To serve as the starting point for my alignment and my analyses. And the next is, there should be some variable regions. And as microbes evolve, these variable regions undergo change, but conserved domains remain same. And I think this is a very good time to mention that the phylogenetic trees are very helpful and very useful because they give us an idea of the evolution. So the reason why we started making phylogenetic trees was because we wanted to know who was our last universal common ancestor, LUCA. And we assume that our last common universal ancestor, or LUCA, had certain genetic fingerprint, and as it evolved, its daughter lineages had slightly different genetic fingerprint, but there were some, and if we could really track these genetic fingerprints and how they changed over time, we can get an idea of which microbes evolved first, came first, and which came later. So let's say we have sampled 1, 2, 3, 4, 5, 6. We know their genetic fingerprints, but we do not know which came first, which evolved later. But now if we make their cluster dendrogram, we notice that 1, 5 and 6 are more similar to each other, 5 and 6 are more different from each other, but they are more similar to 1. So in this case, the similarity between 1 and 5 would be really high, but the similarity between 5 and 6 would be comparatively low. Let us say 0.94, these are just arbitrary numbers, 0.99 similarity between 1 and 5, 0.98 similarity between 1 and 6, and 0.94 similarity between 5 and 6. So this way we can say, because they are more similar to 1, we assume we know that they came from 1. So they branched out from 1 and they are now different from each other. So similarly we notice that the dissimilarity between 1, 2, 3 and 4 is less than the similarity they share from some other organism, 0. And then we can make our evolutionary tree, and now we know that 0 is our last universal common ancestor. So science is still finding out what our last universal common ancestor is, who it was, what kind of microbe it was it bacteria, archaea, eukarya, we do not know, eukaryote we do not know, but we are still making these trees. So how do we make these trees? We look at certain genes of interest. And these genes should have certain characteristics. As evolution goes on, there should be parts of these genes that do not undergo any change. So that is the marker for the gene. Okay, I am seeing this conserved domain of this particular, that is specific to gene A, so this should be part of gene A. And then there should be variable domains, as 0 branches out into 1, 2, 3, 4 lineages and 1 branches out into 5 and 6 lineages. I should be able to say, okay, there are these differences. So it looks like this. These are universal domains, and then there are hypervariable domains where there are for variable domains or hypervariable domains where variations happen. So what we do is, when we are seeing the similarity between 1, 2, 3, 4, 5 and 6, we align them. So this is 1, 2, 3, 4, 5 and 6, so forth. We align their conserved domains and we notice the differences in their, we notice the difference. Here is A, here is G. So we know that, okay, there is a difference here, and then we count how many differences are, and that is how we get our similarity. So these are evolutionary tree, and these are the 3 properties that our gene of interest should have: universality, conserved domains and variable regions. Typically early on, and it is quite a typical practice even now, we found out that there is one gene that satisfies all these 3 qualities for all bacteria, and that gene is 16S rRNA. It is not gene that encodes for a protein, it is part of the ribosome that participates in translation, and it is 16S ribosomal rRNA. So RNA, it is RNA, so it has U instead of T, and 16S, this is when we centrifuge it, depending on where it precipitated, we have given it this name. There are actually 4 components of rRNA, 16S is one of them. So this particular rRNA we know has conserved domains and has hypervariable domains. So people have made beautiful, big, elaborate trees of all microorganisms that we know on basis of 16S rRNA gene. In this particular slide, we were talking of another gene, NIFH. Now this gene encodes for nitrogenase, and I think this is, you should note this, that when we manually write name of a gene, we make sure that we underline it, and when we type it on a computer or typewriter, we always italicize it. So either underline it if you are manually writing it, or italicize it. Okay, so we were talking about NIFH gene, which encodes for a particular part of nitrogenase, which is very important for nitrogen fixation. It's essential. Now it said that when people were investigating different parameters that affect agricultural output and the nitrogen assimilation in plants, and they were looking at how nitrogen assimilation and fixation happens in environment microbiologically, they found out about this gene. What they did was they started making trees according to this gene. So instead of using 16S rRNA, they used NIFH gene, and what we very soon found out was that this is not a very good gene for making phylogenetic trees, because even though it is universal to all nitrogen fixing bacteria, it has more variable regions and conserved domains. There is such variation in different NIFH genes that we cannot make meaningful evolutionary trees. So even though we use this as a marker for nitrogen fixation, if we want to take all nitrogen fixing bacteria and make the evolutionary tree to find out what their last universal common ancestor was, then we need to go back to 16S rRNA gene. Alrighty. Now sometimes diazotrophs will form symbiotic relationship with plants, animals and fungi, which is very important for plants like legumes, and in this case the host will give them a nice environment to live. It will give them all nutrients such as carbon source, energy. It will also regulate the oxygen content, so making sure that they are not exposed to excess of oxygen, and diazotrophs in return will provide the fixed nitrogen to the host. Now the free living diazotrophs, not all of them form symbiotic relationship, by the way. They need a mechanism to protect the diet nitrogenase from oxygen, and the easiest way is don't grow when there is oxygen present. So that is fixing nitrogen only when oxygen is scarce, and the other solution is be obligatory anaerobic. So only grow in under strictly anaerobic conditions. So next we have nitrifying and denitrifying bacteria. Now microbes that convert inorganic nitrogen, nitrate and nitrite to gaseous products are called denitrifiers. They are typically facultative aerobes, they are oxidizing, and they are chemoorganotrophs. Microbes able to live chemolithotrophically by utilizing ammonia nitrite are called nitrifiers. They are typically obligate aerobes, some can be autotrophs as well. No microbe is known to completely oxidize ammonia to nitrate. And thus two kinds of microbes, ammonia oxidizers and nitrite oxidizers, are employed to oxidize ammonia to nitrate. And that is all for today. In the next class, I will be talking about functional diversity of strange microbes. So, we will be talking about microbes that are bioluminescent, microbes that are magnetic and are very strange in many different ways. So, wait for the mystery of mysterious microbes, and this is all for today. Thank you very much.